Hi, I'm Mrs. Murdock, and this is going to be about the Lac Operon. So this is about how bacteria control the production of certain enzymes when there's food in the area, and they want to make sure they don't waste energy making enzymes for food that either isn't there or food that isn't preferred. So if you look at this simple graph here, let's start with this. Let's say that we have a petri dish, and in that petri dish we have some E. coli bacteria growing, and the scientists have added both glucose and lactose to the surface of the agar. So the bacteria have a choice between these two sugars. Glucose, as you know, is probably the preferred sugar of every cell on the planet because of the very evolutionary conserved process of cellular respiration. Glucose, it fits into the first glycolysis enzyme that's floating around in the cytoplasms of every living cell, including the simplest bacteria. So if you're a bacteria cell, your very favorite food in the world is glucose, right? But you don't have the brain to be able to figure out, well, if glucose and lactose are there, go for the glucose because because it's less energy consuming than having to digest lactose. Lactose, a milk sugar, also a very good source of food. But if glucose is the better food, don't waste your time making enzymes for lactose if glucose is present. So first of all, what they found when they exposed bacteria to these two types of foods is that the curve of growth, of bacterial growth over time, went like this. First there was a steep growth, and then there was a plateau where they didn't grow, and then there was another steep up curve of growth. And they wondered about that. Why is it that these bacteria, they've got these two types of food, and there's this bump in the growth? So what's going on? So let's go over to our Lac Operon diagram that you may recall. So here's a strand of bacterial DNA, and this particular strand of DNA has the Lac Operon in it. So you have the structural genes over here where you have X, Y, Z, and they're clustered together for efficiency, so that as long as this promoter is able to bind with the RNA polymerase enzyme here, and the RNA polymerase can move along the strand, you can get those three proteins that will help in the digestion of lactose sugar over here. But as I said before, there needs to be kind of a switch, an on-off switch, so that you can turn this operon on as long as you have lactose present and glucose, which is preferred, just isn't there, right? But then you can turn it back off if glucose shows up, because you want to eat up the glucose first. So how does it do that? Bacteria doesn't have this big, wonderful, marvelous brain that you have, where you can use logic and say, less energy burnt, and I'll get more ATP out of it. I'm going to go for the glucose. It can't do that. So instead, it's more of a stimulus response, molecular binding, not binding type of a thing. So you have upstream from every operon, you have regulatory genes. That's what these are. These two strands of DNA. And there are two regulators for the promoter switch in these operon. One is the repressor, which very obviously, if it's present here on the operator, it's going to turn the operon off. So it will not make the enzymes to break down lactose. So the repressor is controlled by the presence or absence of lactose itself. So if you have lactose, if you squirt milk onto that petri dish, the lactose molecules, these sugar molecules, bind to the repressor, and they cause a conformational change. They'll bind to wherever there are repressor molecules, and if the repressor is bound to the operator, the lactose binding to it will cause it to release and no longer be in the way of the RNA polymerase, which will then be able to go through, and these three enzymes, which are useful for digesting lactose, can now be created, little tangles of amino acids, as you know proteins are, and these enzymes are then going to go and digest the lactose and break it down and put it into those respiration pathways and get some ATP out of it. So that'll be good for our bacteria. And eventually what happens is that sort of regulates itself after a while, because eventually all the enzymes break down that lactose and it's not there anymore, and then it releases off of the repressors and it causes the repressor to go back and repress again, right? So no more lactose, no more repression. No more stopping the repressor, and therefore you stop. You stop everything, and the repressor goes back to repressing, and you don't make any more enzymes because there's no more lactose to break down. That makes lactose into what we call an inducer. Lactose is the inducer of this inducible system. Inducer means something that nudges or pushes or starts something, right? So lactose gets the operon started because it takes that repressor off. Okay, so now the plot thickens because now you have this other activator gene over here, right? That creates an activator protein. So one of the regulatory genes creates this repressor, which is always floating around waiting for lactose to come over it to deactivate it or activate it. And the other regulatory gene makes this activator, which I kind of drew like this green Pac-Man. Both the activator and the repressor are affected by other things binding with them that are either going to make them work better or not work at all. So with the repressor gene, with the repressor protein, those little lactose molecules that I drew there, hold on, erase, will stop it from working and therefore turn the repressor off, thereby allowing the enzymes to be made. But the activator works differently. The activator depends on the presence of glucose. The presence of glucose. So now glucose, instead of being an inducer, glucose, when it's present, is going to be, well, I guess I'd call it a negative or a deactivator, is what glucose is. A deactivator. Because if glucose is there, why waste your time making lactose enzymes? Deactivator, terminator. Okay, there we go. So what happens with glucose is this. Glucose has an effect on the co-activator of the activator. Okay, so stay with me. So when there is high glucose, right, when there's lots of glucose, it causes a decrease in another molecule that you might recognize called CAMP, which is that same secondary messenger that we saw in some of our signal transduction pathways in eukaryotic cells, in hormonal pathways, right? But here in this environment, it works a little differently. CAMP works as a little co-activator, right? So it's like that little slice of pie that fits in right there into the activator. And when it's there, it makes the activator work and makes the enzymes easier to make, allows the RNA polymerase to work more easily. So look at what glucose does. When glucose is high, there isn't any CAMP, so no activation. When glucose is low, it's just the opposite. There's a lot of CAMP. Lots of CAMP. And that little CAMP, little red pie slice, is then going to fit in here. And that's going to result in an activated activator protein. There. Now it's activated. Now that it's activated, it's going to go up here and settle itself on the promoter, kind of like the transcription factors we talked about in eukaryotic promoters, right? So it's a little bit like that. If it's there, it makes RNA polymerase much more likely to bind, right? So it's not just the lack of repressor that has to be here. You also need the presence of the activator to allow this to go and transcribe and translate and create those three proteins that are then going to go and find the lactose sugar molecules that are over here inducing and keeping the repressor away, right? So in order for the Lac Operon to really work, you need two things to be true. You need the presence of lactose, which is an inducer which keeps the repressor out of the way and from binding and getting in the way of the RNA polymerase. And you need there to be low glucose. You need there to be almost no glucose at all, because the glucose being low makes the camp over here, this little red thing, the camp, high, and then causes the activator to then bind to the promoter and make the whole thing work. So there's the Lac Operon. That's the total control. Both the on switch are controlled by two different sugars. So lactose acts as the inducer. Without the lactose there in the first place, there's no reason to make the enzymes, because the lactose binds to the repressor and takes it out of the way, right? And then the glucose needs to be low. You can't have a lot of glucose, because why would you want to activate the Lac Operon if you've got plenty of glucose that doesn't need those Lac enzymes? It has enzymes already in the cytoplasm waiting for it. Okay, so now let's go back to the other picture. So here, now think about this again. Let's draw a little red line on the top of this first upswing of the curve. What is the bacteria going to eat first? What does it prefer? So what's it eating here when it first starts to grow? And will the Lac Operon be active here? Think about that. Which food is it preferring, and will the Lac Operon be operating at this point? Then go up here. What about here? In this upslope right here. What food is it now eating? Must have run out of something, and now it's eating something else. And will the Lac Operon be operating here? Okay, think about that, and hopefully that will help you with your Go Formative today.